Hey, good morning, and looks like we got a fairly good turnout already today, so we're going to go ahead and get started. Anyone have any questions before we start? Okay, and I'm going to go ahead and get started. If you have a question, shoot it out through chat or send me an email. I'll be, I get back to you as quick as I can. So let's go ahead here. And if I remember correctly, we had just finished talking about apoptosis and self-destruction of the cell. That was, let me go back, turn this on. There we go. Last thing we talked about was apoptosis, programmed cell death, self-destruction of the cell. When a cell is so badly damaged, it can't repair its DNA. It tries very hard in the S stage of the cell cycle to fix itself, but it can't. So if it can't fix it, the safest thing for the cell to do is to destroy itself. It releases its own digestive enzymes and dissolves itself, and then other cells come along and finish the job. The macrophages, the white cells, the large the large monocyte cells are turned into macrophages and dissolve it, as well as just the regular whites, like the lymphocytes will do it too. Now, something that we have in our DNA to help protect us against damaging to protect the DNA from getting damaged, because it will get damaged from all those sources we talked about the other day. Ultraviolet radiation is a good example here. If we get a sunburn, regardless of our skin color, if we get exposed to too much ultraviolet radiation, we can damage the living cells in the basement layer of our skin, in the basement layer of the epidermis. If we continue to damage those living cells, then it's entirely possible that we can develop skin cancer. One sunburn is not going to cause skin cancer. But I read somewhere that severe sunburn early in life may actually have some sort of predisposition to skin cancer later on. So haven't seen a lot of work to support that, but it's a possibility. But something we have in our own DNA that protects our DNA is something we call tumor suppressor genes. Now remember, a gene is just the location of a trait on our DNA strand. That's all the gene is. It's the physical location of the code for a particular protein. Now you may have heard the expression, you may have said it yourself, that breast cancer runs in my family, or colon cancer, or lung cancer, or something along those lines. And while that is not a technically correct statement, it is a valid statement. When your patients come to you and say, well, I'm concerned because Uncle Fred has colon cancer and my dad has colon cancer and my grandma has colon cancer, and now I'm concerned. Okay. And then they say, because it runs in the family. Well, okay, that's a valid statement. What really is happening is that particular family unit of the patient you're talking to is genetically predisposed to that. And why are they predisposed to it? Well, we have these things called tumor suppressor genes. Tumor suppressor genes are there to protect us from various cancers when the DNA malfunctions. See, what is a cancer? Cancer is nothing really anything more than uncontrolled cell growth. No checks and balances. Cells will continue to grow and grow and grow over top of themselves, and just without any checks in place to stop the growth. Tumor suppressor genes are designed to repair the damaged DNA that allows that to happen. Cells aren't supposed to be growing, randomly and overgrow each other and form a mass, in an uncontrolled manner. We have very specific start and stop signals in our cell cycle. What the tumor suppressor gene does is it makes proteins that shut down the cell cycle. If the DNA is damaged, when we get to at the end of the G1 stage, when we get to that first checkpoint, if the DNA is damaged, the tumor suppressor gene will stop the cell cycle right there and initiate repairs. That's what it does. It generates proteins that can slide up and down the DNA strand looking for mistakes, because it's obviously, it's detected some error in the DNA. Something's wrong in the DNA, and the tumor suppressor gene will make proteins that literally slide up and down the strand of, slide up and down the DNA strands, looking for errors and then repairing them. Slicing out mistakes, putting in the proper, base sequences, because the base sequences is what determines what kind of protein we're making. That's what the tumor suppressor genes do. If we can't repair damage, then the tumor suppressor gene initiates apoptosis. And so this tumor suppressor genes are scattered throughout our DNA. There's a variety of types here, and they are looking for errors in our DNA. That's what they do. And the most well-known tumor suppressor gene, one that's been studied more often, is called tumor suppressor gene P53, or protein 53, or we just refer to it as the P53 gene. And it controls and protects us against a wide variety of cancers. Now, if it works, why do we get cancer? Because the P53 gene may mutate on its own. It too may mutate or experience a mutation. And when it does, the cancer that is the P53 gene protects you against, when there's a mutation to the P53 gene, the particular one of the types of cancers that the P53 protects you against then has a greater chance of developing. And the P53 protects us against a wide variety of cancers. And if there's a mutation, it doesn't mean you're going to get all those cancers at once. You may never get them. It just means that now you're at a greater risk. But when the P53 gene detects damage to the DNA, then the P53 gene will generate these special proteins and special enzymes that slide up and down across the DNA strand and slicing out the mistakes. So this is what a tumor suppressor gene does. It can repair the DNA, and this is a great thing. We don't know how often our DNA is damaged in our cells. We're not aware of it because our tumor suppressor genes are fixing any problems. It probably happens all the time. We're probably constantly exposed to a variety of viruses and pollutants and sunlight, ultraviolet radiation, maybe ionizing radiation, chemicals in our food additives, for example, pollution in the air and the water by chance, exposure to smoke of any kind. There are a wide variety of things that can cause mutations. And the tumor suppressor gene works to repair those mutations. So we go through life if we don't ever experience any kind of cancer, it's because our tumor suppressor genes have done a very good job. It's not because we're never exposed to anything, because we likely are. But the tumor suppressor gene is protecting us. So what happens with the gene is it has three options, essentially. Three things it can do, of course. The first thing, of course, is it stops the cell cycle at the G1 stage. Interphase is still going on in the background, but we're not going to go to the S stage where we replicate our DNA. The first thing a tumor suppressor gene can do is initiate repairs to damaged DNA. You know, it can slice and dice out the mistakes, the errors caused by mutations, and only mutations. It doesn't do anything for genetic issues. But a mutation that occurs, the tumor suppressor gene will stop us at the G1 stage, fix the damage, and then allow the cell to go on. It's the first thing that can happen. If it can't fix the damage, then the P53 gene will trigger apoptosis. Too much damage, can't fix it. The third thing it can do is what we call senescence. Senescence is essentially allows the cell to go dormant. It won't grow anymore. It will essentially shut down. And the driver for this is the presence of what are known telomeres. Now, telomeres are little structures, they're made of nucleotides that are found at they're non-coding sections of the DNA. They don't code for anything, at least we don't think so. Telomeres form caps on the ends of the DNA strands. Now, if you have shoelaces, you know what the plastic cap is on the end of your shoelace. That's a little thing that allows you to run the shoelace through the opening in your shoe. And you know what happens if the plastic cap comes off, you know how hard it is to get your shoelace through the little hole. We all know that. Okay, that's what a telomere does. A telomere looks like that little plastic cap. It's attached to the end of the DNA strand. Looks just like what we have at the end of our shoelaces, if you have shoelaces. Anyway, the point is, now imagine every time you tied your shoe, that little cap got shorter and shorter and shorter. It wore away, and eventually it would be gone. And you couldn't use the shoelaces anymore because the caps were gone. They would no longer stay in the hole, and you couldn't tie them anymore, and they wouldn't be any good. Well, this is what happens, or at least we think what happens in our DNA. Every time a cell undergoes mitosis and cytokinesis, it's thought that a little bit of those telomeres are removed. And as the cell goes through multiple replications over time, more and more of those telomeres are worn away. That's Monday, I can't talk. The telomeres are worn away. Once they get to a certain length, or a certain shortness, the cell doesn't undergo mitosis anymore. And that particular cell, that's the cell line from that cell, stops. No more replications. Does that mean, does that mean it's dead? Well, it's thought that it goes into, it stops, it degrades, it may go dormant, it may go into senescence. Particularly if the DNA has been damaged, the P53 gene will often tell the cell, hey, you've been around a long time, your telomeres are short, we're not going to replicate anymore. This is the end of the line. You go dormant from here on out. If there's damage, it doesn't go anywhere. The cell is now dormant. It goes into senescence. It goes into a state of suspended animation, if you will. We think that the telomeres also may have something to do with regular aging of our bodies. because if the cells stop growing and changing, then the things that that cell was doing stop. Now, we have billions of cells. One cell stopping isn't going to hurt us. But a lot of cells in that line, gradually it accumulates. We don't repair damage as well. Our bones might get a little weaker. We don't make as many enzymes like we need, or the concentration we need, whatever the cell does. Maybe we're not producing everything we need, as we age. So it may have to do, aging may have to do with telomeres wearing away. And it's interesting that cancer cells, which cancer, of course, is nothing but uncontrolled cell growth, cancer cells never have short telomeres. They reproduce and reproduce and reproduce, and they copy themselves over and over and over again. And it's just this random explosion of growth, and their telomeres don't shorten at all. Their telomeres stay fully active. Because if the telomeres of a cancer cell were to shorten after each division, eventually the cancer would stop growing and die out. But that's not the case. Anyway, back to P53. P53s, if they mutate, then you are more likely to develop a cancer that the P53 gene protected you against. And about 60% of all cancers that occur are due to a mutation of one type or another in the P53 gene. Now, the P53 gene covers a multitude of cancers like breast cancer, lung, liver, skin, prostate, bladder, cervical cancer, and colon cancer. So does a failure of the P53 gene due to mutation set you up for all of those? No. Usually it's going to cause an occurrence of one type of cancer or another. For example, breast cancer. Breast cancer, probably the number one or number two cancer. I think it's number one cancer in females, is often associated with a failure of the P53 gene. It also causes breast cancer in males. Males can develop breast cancer. The cancer rate is usually 1 out of every 350 cases of breast cancer occurs in a male, because the breast cells in males and females are nothing more than modified sweat glands. So they have the same basic underlying structure. The cells in the breast, the underlying structure in the male doesn't seem to be functional like the female breast cells are as far as producing milk and colostrum and stuff like that. But still possible. Even though the occurrence is much lower, it still occurs. So 60% of all cancers, addressing all these different categories here, colon cancer, cervical cancer, bladder cancer, prostate cancer. So if a patient says that a particular cancer runs in their family, it's usually because of the genetic predisposition of the P53 gene to fail in its protection for that type of cancer. Prostate cancer. If your uncle had prostate cancer and your grandfather had prostate cancer and your dad had prostate cancer, you got a pretty good chance of developing prostate cancer yourself if you're a male. So you have a fairly good chance of that. So you have, your patient, your male patients, because of a history, need to be screened more frequently. The same with breast cancer. If your patient has a family history of breast cancer, crazy Aunt Sally and Uncle Fred and their grandma and their sister and 3 female cousins, are they going to get breast cancer? Maybe. The odds are greater because of that. You know, doesn't mean they're absolutely going to get it. No, there's not 100% guarantee they're going to develop any kind of cancer, but they have a greater chance of it because it appears that their P53 gene usually, fails more frequently than other types of cancer. Now, of course, then there are the breast cancer genes, the BRCA1 and BRCA2, and we'll talk more about them in AP when we get into reproduction in AP2. We have a tumor suppressor gene that does affect prognosis, survivability of breast cancer, and that is the P27 gene. The P27 tumor suppressor gene does inhibit the growth of the cancer cell. And as long as the P27 gene is there and doing its job, while the breast cancer may have developed, the prognosis, the survivability it stays high. So breast cancers are a funny type of cancer because they occur at different rates. They're not easy to pin down. It's not funny, haha type funny. It's funny in that it's different. Some breast cancers will grow rapidly. Some will grow very slowly. Some breast cancers only grow on the surface of the breast. And we'll talk all about that in AP2. But it's hard to pin it down and say, well, every breast cancer is going to follow this pattern because some do and some don't. So prostate cancer in males is the same way. Some prostate cancers in males grow rapidly, and some grow so slowly that the conventional treatment is to do nothing for quite a while. So anyway, P27 gene is the other gene that we pay a lot of attention to. Again, these genes can fail. The whole thing doesn't fail, but enough of it fails to affect a particular type of cancer. So what the P27 gene does, and what the P53 gene does, as an example here, it will detect damage to the DNA. It has two choices then. It can stop the cell cycle and repair the DNA and start it back up again. Or actually, I guess it's three choices because it can initiate senescence also, which again is essentially the cell cycle stops at senescence. Nothing happens. You can either stop the cell cycle, repair the damage, and start it back up again, or you can trigger apoptosis and destroy the cell. Now, we've all seen examples of apoptosis in action, protecting our cells. Anyone that's ever experienced a sunburn, anyone that's ever seen anybody with a sunburn, one of the consequences, regardless of skin coloration, one of the consequences of a sunburn is this phenomenon known as peeling. Skin peels. And what that peeling is nothing but dead cells. Now, what's so unusual about that? Well, normally when cells die, and they die all the time, every time you rub your hands together, you're shaking off dead skin cells. But you don't notice it because it's not in such a large volume. Just sort of like this random distribution of dead skin cells. However, when an individual who's experienced a sunburn, with a sunburn, ultraviolet radiation from sunlight penetrates through the epidermis to the living cell layers, in the basement layer of the epidermis, where that's the first layer we encounter living cells. We know that normally living cells are filled with keratin, are pushed up and they die and then they slide off, and they're there to protect the outer layers of our skin, and that's fine, that's what they do. But if you expose these living cells to unusual amounts of ultraviolet radiation, it you lay out in the sun at the beach all day. And, famous words of many people, oh, I never burn, and then they have this extraordinarily painful burn, because what happens is the ultraviolet radiation penetrates to the basement layer of the epidermis and damages those cells. It damages the DNA in their nucleus. And the P53 gene says, hey, I can't fix this. So we have this massive die-off of living cells all at once. And you see skin peeling off your patient's arm because they've been exposed to too much sun, too much ultraviolet radiation. You don't see that phenomena any other time, this peeling of the skin, because we shed cells, say that quickly, all day long. But when there's a massive kill off of cells because the cells have been exposed to ultraviolet radiation, the P53 gene says, I can't fix this, initiate apoptosis, all those cells die and they're pushed up and they all come off in sheets. There's thin sheets of skin that you see after a person's had a sunburn, a very painful sunburn. That is apoptosis in action. The P53 gene fixed that problem, because otherwise those cells that if apoptosis wasn't initiated, since all that DNA was damaged by ultraviolet radiation, there's a much greater risk for skin cancer there. And that's why those cells initiate apoptosis, to get rid of them, to take their damaged DNA and be removed. Now, we still don't know why, if every cell got releases. Obviously, if P53 gene fails, then those cells that became cancerous, or had damaged DNA could become a skin cancer. So, okay. So the P53 gene does a really good job in protecting us on all these things that we're exposed to. It's only if it fails or it mutates that we have an issue. And even then, there's no guarantee that just because the P53 gene has mutated that we're going to develop a cancer. We don't even know that the P53 gene has failed, and we may never develop a cancer. Just because there's a family history doesn't mean that your patient's going to be the one to develop a cancer. It means that you have to, as a healthcare provider, you have to watch them more frequently. They have to watch themselves more frequently. Okay. So let's move on and talk about how the DNA does what it does. We've been saying all along that DNA is our genetic code, and that the genes are the physical locations of nucleotides on the DNA strand. Now, remember what a nucleotide is. A nucleotide is a phosphate attached to a 5-carbon sugar attached to what we call a nitrogenous base, a base made with nitrogen involved there. So that's a nucleotide. Nucleotides are the units that we use to make up a DNA strand and an RNA strand. The nucleotide bases, the bases at the end of the nucleotide, the order of those bases showing up in the DNA is very, very important, because the sequence of the bases dictates the order of the amino acids that show up in a protein. And the order is very important. Proteins are made of amino acids. There are only 20 amino acids, and yet we make an infinite variety of proteins. I mean, consider a dictionary. A dictionary has about, 300,000 words in it. And there are only 26 letters in the English alphabet. So 26 letters can make 300,000 words very easily. It's just how they're arranged. And the order of the letters in a word means something. And it's very important, on how the word is assembled. It may have a totally different meaning if you change the order of the letters in a word. So we won't even get into different pronunciations of words too, but that has nothing to do essentially with DNA. The order of the bases in the DNA dictates the order of the amino acids in the protein. And just like in words, the order of the amino acids is very, very important. An amino acid put in the wrong place can create a problem. So it's very important that we have a sequence that we follow. The sequence of the bases in the DNA, the adenine, the guanine, the cytosine, and the thymine, the order of those bases dictates the order of the amino acids. Consider this. Hemoglobin, a fairly large protein, 360 amino acids long. You've probably heard me say this before, and you'll probably hear me say it again. But hemoglobin, 360 amino acids long. The number 6 amino acid in hemoglobin is glutamic acid. Okay, that's fine. If glutamic acid is not in the number 6 spot, but valine, the amino acid valine is placed at the number 6 spot. So for whatever reason, you take out the number 6 amino acid in this 360 amino acid chain. You take out the number 6 amino acid, which is glutamic acid, and you put in valine. You still have your protein. You still have hemoglobin, but now it doesn't work right. Now it becomes twisted and brittle and bent, and it's shaped like a crescent, and it shapes the red cells to look like a crescent. And now you have sickle cell disease in your patient. Sickle cell disease instructs the gene for hemoglobin to always put valine in instead of glutamic acid. When blood cells are being made, the DNA dictates, okay, here's how we're going to make our hemoglobin. The instructions for the hemoglobin, the gene for hemoglobin in a person with sickle cell disease will always say, put in valine at the number 6 spot instead of glutamic acid. And that makes the hemoglobin, it reduces its oxygen carrying capacity by 80%. It twists the hemoglobin around to form a crescent shape, and it causes the red cells to assume a crescent shape or a sickle shape. That's where we get the sickle cell disease term from. One little mistake, the wrong amino acid in the right spot. Glutamic acid and valine are 2 different amino acids. The consequences are devastating. So the order of the amino acids is very, very important. Their order is dictated by the order of the bases in the DNA strand. So that's what a gene is. A gene is made up of nothing but a long strand of nucleotides. 10,000, 20,000, whatever the length of the protein that needs to be made from that gene, is going to be a sequence of bases that specifies the order of the amino acids. Each cluster of bases is unique for a specific amino acid. So it's like a cookbook. It tells you the ingredients and the order you have to add them in. The order you have to add them in. You wouldn't take a cookbook and follow instructions on how to make a biscuit and take and change the sequence so that you take the flour and put it in the oven at 400 degrees before you've added in the eggs and milk and butter and salt and whatever else you put into a biscuit. The sequence of instructions is very, very important. So the sequence, a gene then is nothing more than all the nucleotides, all the bases on the nucleotides collected in one spot to control the production of one particular protein. And we've got hundreds of thousands of genes throughout our body because they control hair color, eye color, skin color, body stature, protein enzymes and hemoglobin and muscle fibers and antibodies. Also, everything that makes us us is contained in the DNA instructions, at least our physical appearance anyway. So everything that we have to make is directed by the DNA. So and it's very important that we follow those instructions. So that, so that we have genes for everything. Do you know that the ability to, whether or not you have dangling earlobes or attached earlobes is genetically controlled? The ability to roll your tongue, this is a hard-hitting genetic trait, is a genetic trait. Whether or not when you put your hands together, if your left thumb goes over your right thumb, that's a dominant trait. I don't know what benefit it has, but that's a genetically controlled trait, as well as more important things like hemoglobin production and what type of hemoglobin, and blood typing and antibody production, little things like that that are probably a little more important than whether or not you can roll your tongue. So anyway, the DNA, the genes are, the DNA strand has all of this information. Every cell has the same information. Every cell has DNA in their nucleus, except red cells. Red cells don't have a nucleus. They kicked it out during their development so they could put in more hemoglobin. So they don't reproduce. At least the red cells, once they're formed, don't reproduce. Every cell has DNA. Only the sections of the DNA that are in use for that cell are turned on. If you're a skin cell, the DNA for being a skin cell is turned on, but the instructions for a liver cell are turned off. But they're there because everything's there. Within the gene itself, not everything is a coding section, meaning the instructions that are on the gene, not everything is used. There are areas of a gene that are called non-coding regions. We call those sections introns. Introns. Scattered throughout the gene, they're going to be bases, nucleotides that don't appear to do anything at this point. We call them non-coding regions. We used to call them junk DNA because we didn't know anything about them. We didn't know what they did. Now we know that they are important, but we don't understand yet why they're important. So we refer to them as non-coding. And the reason we know they're non-coding is the other section of the gene, the section of the gene that does the coding is called the exon. And how do we know it's the coding section? Because when prior to issuing instructions to make a protein, the information on the gene is copied, and the non-coding sections, the introns, are sliced out of the copy and removed, and the exons that are left are glued together. Now, not in the DNA, but on the RNA. So we take the information that's important, at least right now, that's important for making the gene, making the protein. Take that out of the copy the whole gene, slice out the non-coding regions on that copy, and save what's left. You know, if you have copied grandma's recipe for biscuits that she got from her great grandma, so the recipe's well over 100 years old, maybe even more. And there are instructions in the recipe on how to heat up the wood stove to bake the biscuits. Well, those instructions haven't been used for probably 80 years or more, but they're still there because they were in the original recipe. Now, obviously, you could skip over that. In fact, if you wanted to, you could, on the copy that you have, you could just cut that out and not bother with it anymore. That would be removing a non-coding region in the recipe, because most of us don't cook on wood stoves. I say most, because there's probably some people that still, for one reason or another, like to cook off a wood stove. Anyway, that's beside the point. Okay, so we take out the non-coding segments and leave behind the coding segments. Protein synthesis is how we make new proteins all the time, the building of new proteins. The code, the DNA code, as I've said, is based on the order of the bases adenine, guanine, cytosine, and thymine. In the DNA strand, in the gene on the DNA, these bases are collected together in clusters of 3 called triplets. The triplet code, the sequence, the order of the bases in the code dictates which amino acid is going to be used. So, for example, if you have 3 bases together, you have guanine, guanine, and cytosine. That's a triplet. That codes for the amino acid known as proline. If you take guanine, cytosine, and cytosine, the first one was guanine, guanine, and cytosine, and that gives you proline. If you take guanine, cytosine, and cytosine, one little change here, but significant, it codes for the amino acid arginine. So the order of the bases within the triplet determines which amino acid is going to be used in that protein. Each triplet codes for a particular amino acid.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA each compose 4 different nucleotides, which differ in their nitrogenous bases. Three of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 length-ranging atoms, called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbohydrates and nucleotide sugars are linked together with the proteins bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The outer ring is bound to the nitrogenous base, the smooth ring carbon to the next nucleotide, and the fine ring carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are often similar to fast-phase and 3-phase reactions. The 2-phase break determines the 3-phase position of the nucleotide, and the 5-phase at the 5-phase position of the next nucleotide. A measuring instance of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotides. However, there is the need to invest in just
Yeah, the order of the bases is very, very important. It is extremely significant because the order of the bases within the triplet tells you what amino acid that has to be assembled. And if the order is wrong, or if there's an error in the order, then you're going to put in the wrong amino acid in the protein assembly. So, and that's very critical to get that right. Now, DNA never leaves the nucleus. DNA stays in the nucleus all the time, but it uses RNA to take its instructions out to the ribosome. The ribosome out in the rough endoplasmic reticulum, remember it's called rough because it's studded with ribosomes. Now, the ribosome is the site of protein synthesis or protein assembly. The instructions are sent out to the ribosome on a strand of RNA called messenger RNA. The messenger RNA is a complementary copy of the gene. The only difference is that we use uracil in place of thymine. So if the DNA strand has adenine as a base, the complement to adenine is normally thymine on DNA, but it would be uracil on the RNA. The other difference is, and this is not a critical difference, is that RNA is a ribose sugar and DNA is a deoxyribose sugar. What's much more important is the fact that the RNA is going to be a complementary copy of the DNA. So if the DNA is adenine, the RNA copy will be uracil. If the DNA is thymine, the RNA copy will be adenine. Cytosine to guanine, guanine to cytosine. We'll have the messenger RNA is going to be the complementary copy. So there are three types of RNA. Messenger RNA, which copies the information off the gene and brings it out to the ribosome. The ribosomal RNA, which forms the shape of the ribosome for the protein assembly, and transfer RNA, which brings the amino acids to the ribosome in their correct order. Messenger RNA, like all the RNAs, it's going to be a single strand. It is copied off of the DNA. It's a complementary copy. The DNA strand, the gene is the complement. The gene then the messenger RNA is copied off the gene using the gene as the template. The messenger RNA is the complement to the gene. So it is a complementary copy. This process is known as transcription. And we maintain this triplet code in from the DNA. So we have the complementary copy of the bases in triplet form. In RNA, we call it a codon. Call it a triplet in the DNA and a codon in the RNA. Ribosomal RNA is the structural basis of the ribosome. And then we have the transfer RNA, which is specific. Each transfer RNA is going to be specific for an amino acid, and it will bring each amino acid to the ribosome to assemble the proteins. Now, within the we know that the messenger RNA will have a section known as a codon. The codon are the triplets. The transfer RNA will have an anticodon that turns out is the complement of the codon. So I'll get to that here in a second too. It's a protein synthesis, two parts to it. Transcription takes place in the nucleus. Translation takes place out in the cytoplasm in the endoplasmic reticulum. Transcription is making a copy. You write it down, you transcribe, you take the information off the DNA and you make a copy of it. You know, you're going to copy grandma's biscuit recipe. And you make a word for word copy of it. See, because grandma won't let you take her cookbook out of the house. She keeps the cookbook locked up, but she will let you write down the instructions on how to make biscuits. So that you have made a transcription, you've transcribed the copy. You've made a transcribed copy of her instructions. That's what transcription is all about. You copy the information from the DNA. But bear in mind that when you're doing it from DNA to RNA, the RNA is a complementary copy. You know, if the DNA says adenine, then the RNA has to say uracil. If the DNA says cytosine, the RNA has to say guanine. Translation then uses the information on the messenger RNA to make the protein. So transcription starts in the nucleus. Remember, the DNA never leaves the nucleus. All of the nucleotides that are needed are already assembled inside the nucleus. All the nucleotides needed to make the RNA. At the beginning of the gene, there is a section called the promoter gene. That promoter is only on the DNA side. We don't copy the promoter. We only copy what's after the promoter. At the end of the gene, there's a terminator code. It says stop. We have to open up the DNA. Remember, the DNA strand's a double helix. We have to open that up, open up the helix. Remember, like we did for semi-conservative replication. We open up the DNA strand. We use helicase to get it to open up. We expose both sides. One side is the gene that's going to be copied. You know, there may be, you know, it's easy to conceive of a gene on each side of the strand that's opened up. The side that we're going to copy is the coding side. And we use RNA polymerase. So remember DNA polymerase, we use that to assemble the DNA strands in semi-conservative replication. Here, we're going to use RNA polymerase to make the messenger RNA copy of the coding section of the DNA. We're going to copy our gene, and we're going to use RNA polymerase to assemble it. So the first step is initiation. Gene separate, the DNA opens up, and RNA polymerase starts making complementary copies, starts assembling the complementary nucleotides in a strand. So initiation, we start at the promoter gene, and the DNA is opened up. Now we have elongation as we keep adding the complementary RNA nucleotides. Notice that, you know, we have, if we have adenine here, our complement has got to be uracil. So we are busy making our complement. Our coding, if our coding strand is down here at the bottom, there's our complement to it. Every time you get an adenine on the coding side, you get a uracil on the RNA side. The RNA strand is in red. Not really, but on the animation here. So we have initiation. We open up the DNA strand. The RNA polymerase starts to bring in the RNA nucleotides, and we start building a complementary copy of the coding section of that of the DNA. Yeah, whatever side we're going to copy is the coding side. So the gene is down here in this part. So there we go, making our messenger RNA copy of it. And when we're done making the copy, we stop. There is a termination code that says, okay, stop. Don't go any further. There's another gene next door. We don't want to copy that gene. We only want to copy this gene. So we get to the termination code and we stop. Initiation, elongation, and termination. But we're not done because we've copied the introns and exons. So we have to slice out the introns and glue the exons together. So we use these enzymes called spliceosomes. Not making this up, this is the name, spliceosome. A spliceosome cuts out the introns and then glues the exons that are left together, and your finished product is an all coding segment. And you've taken out the non-coding regions, and all that you have left is the coding regions. And don't know what happens to the sliced off introns. They probably get broken apart and reused. You know, we don't know what the introns do anyway. But I suspect that at the very least, the nucleotides are removed and reused again in making other strands of messenger RNA, because the cell will recycle the nucleotides, because the nucleotides, they don't matter. I mean, the individual nucleotides, why destroy them? They're going to be reused again and again, because all you're doing, all that's important about the nucleotide is just the sequence of the bases. So transcription, we end up our finished product with a sliced and diced spliceosome treated series of exons linked together. And now we have our perfectly copied perfectly copied information off the gene. Use it with only the coding regions to assemble the protein. And then that leads us into translation. Now that we've got our genetic information, we got to be able to assemble the protein. And the messenger RNA leaves the nucleus. It goes through a nuclear pore and goes straight to the rough ER, because the rough ER is continuous with the nucleus through the nuclear pore. The messenger RNA leaves the nucleus and goes straight to a ribosome in the rough ER. And the information on the messenger RNA now collected in codons, clusters of 3, the codon is the counterpart to the triplet on the DNA. The codon specifies the order of the amino acids. Now, a couple things about these codons and triplets. Each triplet on the DNA has a complementary codon on the RNA. Because you have 3 bases on the DNA, that's a triplet. If it's adenine, 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 its complementary copy on RNA is uracil, uracil, uracil. So the codons are going to code for the amino acids. How many codons are there? Well, there are 64 possible combinations. You have 4 different bases, adenine, guanine, cytosine, and uracil, arrayed in clusters of 3. And that works if you take 4 bases to the 3rd power, you get 64 possible combinations. Trust me in this. 61 of those are going to code for an amino acid. 3 of them are stop codons. Now there are only 20 amino acids and 64 possible combinations. It means that you have a lot of redundancy. There's a tremendous amount of redundancy here. So that even if mistakes happen, it may not matter, because the mistake may still code for the same amino acid that the correct version did. So we protect against transcription mistakes by having a great deal of redundancy. Now this chart, and I will confess, the first time I ever looked at this, I had no idea what it was trying to show me. But what this does show is all 64 possible combinations of the nucleotides in the RNA. And you can determine what amino acid you're going to get from the sequence. The codon has 3 bases. It has 3 letters in its codon. Each letter represents the base. A for adenine, G for guanine, C for cytosine, and U for uracil. The order of those bases in clusters of 3 tell you what the amino acid is. If you look on this side, on the left-hand side, where it says first letter, you have guanine, adenine, cytosine, and uracil running up the left-hand side. The left-hand column is guanine, adenine, cytosine, and uracil. This row across the top says second letter, and it has uracil, cytosine, adenine, and guanine here. And over here on the far right side, the column has, each block in the column has uracil, cytosine, adenine, and guanine all the way down. So if I just take a, let's say cytosine, guanine, cytosine, just picking a combination, cytosine, guanine, cytosine, and I want to see what amino acid that's going to give me. So the first letter I said was cytosine. So over here is, go to this the first column here, and there, this is the row for cytosine. So I go across. Now my second letter was G. So I look up across the top here, and I see that G is the last column. So I come down here to the intersection of the G column and the C letter. I said cytosine, guanine, cytosine. So cytosine, I go to this row, and I go across till I line up with the G column, and that's this last one here. And I said cytosine, guanine, cytosine. So I look at the C over here and discover that cytosine, guanine, cytosine codes for arginine, as does cytosine, guanine, uracil, cytosine, guanine, adenine, cytosine, guanine, and guanine, and adenine, guanine, adenine, and adenine, guanine, and guanine. So there are 6 different possible combinations for arginine. Arginine must be a pretty popular amino acid. If I pick another one, adenine, cytosine, uracil. Adenine, guanine, adenine, cytosine, uracil. So the first letter is A over here on the left-hand side. The first letter is A. There's adenine. It is in the third row. Adenine, cytosine is the second letter. So this is the column for cytosine. And the third letter was uracil. Adenine, cytosine, uracil. And that's the code for threonine, along with everything else in this particular box. So there are, whatever combination. Most of them are going to have 2. Phenylalanine has 2 codons. Leucine has 6 codons. Isoleucine has 3. Methionine only has 1. Methionine is always the start codon for our protein. Valine has 4. Alanine has 4. Proline, serine, and threonine all have 4. Tyrosine has 2. Cysteine has 2. Tryptophan has 1. tryptophan. Um, the mistaken belief that tryptophan levels in turkey make you sleepy after you eat. It's not true. So it's an urban legend that the high levels of tryptophan make us sleepy. What makes us sleepy after a big meal is the rise in blood sugar. Not, after a big, if you've eaten a lot of turkey, say at Thanksgiving, for example, and you get sleepy afterward, it's not the turkey. It's it's the carbohydrates that came with it. Anyway, so we see, histidine, glutamine, asparagine, lysine, aspartic acid, glutamic acid, all have 2 codes. Lysine has 4, arginine has 6, serine has 2. So there are only 2 amino acids that only have one codon for it. A lot of them have 4, several have 6. So we have a tremendous amount of redundancy. If you take any 3-letter combination of those bases, you can determine what the amino acid is. This is, this is a nice little tool for determining, and is it going to be relevant later on in your career? Well, I've never been approached in the street and say, hey, what is the amino acid for uracil, guanine, uracil? It's never happened. I'm still waiting, but it's never happened. Anyway, a couple highlighters here. Methionine is the initiation codon. The sequence of the codons always starts with methionine. This is the start codon. One of these 3, UAA, UAG, or UGA, are stop codons. UGA, for example, if you like the University of Georgia, or you like their bulldog named UGA. That's a nice way to remember that. The stop codons stop the production of the protein. So how does this work? Well, here's where we bring in the transfer RNA. The messenger RNA will leave the nucleus. It will go out to the ribosome. It will move into the ribosome. The ribosome has 2 parts, a small subunit and a large subunit, and they will bind on top of and underneath the messenger RNA. They look very much like a big hamburger bun. And the messenger RNA will start moving through the ribosome, or the ribosome slides across the messenger RNA. It doesn't really matter. Either way, we start exposing the codons on the messenger RNA. The codons are going to become binding sites for the amino acids. And this is where transfer RNA comes in. Transfer RNA shaped, different books show it different ways. Some look like a fish hook, some look like sort of like a guitar shape. The anticodon is made of nucleotides. It has a head and a tail. The tail has, or stem has the amino acid attached to it. Up here at the stem end of the or the top of the fish hook, because I've seen it drawn all sorts of ways. This stem region is where the amino acid's attached. The anticodon down here, this region, is the head. Now the anticodon on the transfer RNA will be complementary to a codon on the messenger RNA, so they can bind together temporarily. So if the codon on the messenger RNA is uracil, 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 the anticodon will be adenine, adenine, adenine. Remember, they're complementary. For example, this anticodon is uracil, adenine, uracil. So the anticodon, that's the anticodon. The codon it would land to is adenine, uracil, adenine. Remember, they're complements. Adenine to uracil, uracil to adenine. The anticodon is the complement to the codon on the messenger RNA. So the transfer RNA has the amino acid at one end and an anticodon region at its head that is going to be a complement to one of the messenger RNA codons. The anticodons will only bind to the codon that they are complementary to. Guanine, cytosine, guanine will not land to adenine, adenine, adenine. Yeah. Uracil, adenine, uracil will not land to cytosine, guanine, cytosine. They will only bind to their complement. So when they're bringing in their transfer, the transfer RNA is bringing in its amino acid. The anticodon will look for its complementary codon. And just like in transcription, we have initiation, elongation, and termination. With initiation, the ribosome forms around the messenger RNA. The first amino acid brought in place here is methionine. It lands at the starting position on every messenger RNA. So we bring in methionine. The anticodon for methionine is UAC. The initiator spot, the start codon on the messenger RNA is AUG. Uracil on the anticodon binds to adenine on the codon, on the start codon. Adenine on the anticodon binds to uracil on the start codon, and cytosine on the anticodon binds to guanine on the start codon, right there. So it's always methionine. Now the next one that would come in, the codon is GUA. So the anticodon would be CAU. There's our start codon right there, AUG. There comes the ribosome. There's the small unit. There's the transfer RNA. There's the anticodon right there. There's the large subunit of the ribosome. Here comes a second transfer RNA. CGC, its complement was DCG. We have a second amino acid brought in. They join together. The first transfer RNA goes off. Now we have a third one coming in. The second one goes off. You'll notice that the amino acids are linked together by peptide bonds. Let me go back and show you that again. So what we're going to see here is methionine land in the first spot, and I think it's arginine lands in the second spot. So this is going to be UAC on the transfer RNA. This is going to be GCG, bring in the second transfer RNA. Methionine and arginine are going to join by a peptide bond, and the first transfer RNA will be let go. There it goes. The messenger RNA will slide a little further through the ribosome. A third one comes in. This is lysine. They bind together with a peptide bond, and this process will continue moving down through the messenger RNA until we hit the stop codon. Elongation continues until we reach a stop codon. And we keep sliding down and forming peptide bonds between the amino acids until we reach the instructions say stop. And that's termination. When we hit the stop codon, like in this case UGA, then the messenger RNA and the ribosome break apart. The transfer RNAs go back and reload whatever amino acid they're supposed to be carrying, and now then our protein gets released. And so we see there goes our protein. once we hit the stop codon, the protein is assembled, fully assembled, and it is put into a transport vesicle and taken to the Golgi apparatus for continued processing. Now, the thing to remember here is that we're taking the information directly from the DNA. So what do? We make a complementary copy of the DNA segment onto messenger RNA. Remember, the complement is the opposite. But the transfer RNA has the anticodon on it. The anticodon is the sequence of the DNA. So if the DNA sequence was adenine, 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 the messenger RNA complement to adenine, adenine, adenine is uracil, uracil, uracil. The transfer RNA bringing in the anticodon. What is the anticodon to uracil, uracil, uracil? AAA, which is exactly the sequence you had in the DNA. So the anticodon brings it all back around. So whatever the sequence is in the DNA is replicated in the transfer RNA to bring the right amino acid in place. Mistakes happen. The most common type of mistake is a substitution mistake, where we put the wrong DNA, wrong base in at the right spot. Yeah. Here we see the original DNA sequence is CGA, ACC, CGA. But here we see that in a substitution in the DNA causes a cytosine to go in place of the guanine. So the original strand has been changed. Now, is that going to be critical? Maybe. We don't know. Or is it just going to be, a couple things can happen here. It could be, can produce a dangerous mutation. It can produce a nonsense mutation and the protein is useless. Or it can be non-essential, that the sequence may code for exactly the same amino acid as it did before, because after all, some of those amino acids have 6 different codons. Now here's, hemoglobin is a substitution mutation that is pretty significant. Now remember, this is genetically inherited. It requires recessive traits from both the mother and the father to have hemoglobin, have sickle cell disease. Normal hemoglobin has glutamic acid in the number 6 spot. Sickle cell disease hemoglobin, called hemoglobin S, has valine in the number 6 spot. Very similar structure, but devastating consequences. Sometimes it is tremendously significant. Other times it doesn't matter. Sometimes it's just going to produce what we call junk DNA, or it'll produce not junk DNA, but junk protein. The other possibility is a frame shift mutation. A frame shift is where we cut out or we add a pair of nucleotides in the DNA, and everything shifts down or up, completely altering the sequence of the triplets in the DNA. Because here we have, GCT, AGT, those are matched. The third codon, third triplet in the DNA should be ATA, but by inserting guanine and cytosine, now our third triplet is GAT, and everything here slides down one. If we slice this out, we sliced out one of these, the same thing would happen, only we would have slid up one. Frame shift mutations are extremely dangerous because they destroy the gene. And that is essentially the end of the end of chapter 3. So it's 10:59, so I'm going to get us out of here. We've finished chapter 3. On Wednesday, we're going to start the chapter on metabolism. It is the next one in the sequence. In the content section, it used to be called chapter 24, based on the old book. If you have in fact, most textbooks, I think even the OpenStax book will follows that pattern. So anyway, so we will look at metabolism on Wednesday. Lab this afternoon, if everything works okay, we'll be looking at the heart again. So, okay, I'm going to get us out of here. Not your lab. I'm sorry. I was thinking AP2. Don't mind me. It's Monday. So, okay, I'll see you all later.